Welcome to today's deep dive.
Uh,
And I have to say, the mission for this one is pretty incredible
it really is, yeah. We are getting into something totally fascinating today.
because, you and I and, well, everyone listening, we tend to walk out our front doors every morning and just operate under this very comforting assumption.
Right, the assumption that the world is a finished map.
Exactly. It's like an encyclopedia that has already been, meticulously printed, bound, and just placed on a shelf.
Yeah, you look at a towering tree on your street and you just say, oh, that's an oak.
Right. Or you look at a weed sprouting stubbornly in the cracks of the concrete and you go, okay, that's a dandelion.
We rely heavily on those labels, don't we?
We do because, they make our environment feel predictable, safe, even.
Yeah, it's a psychological shortcut. We like to think the era of grand biological discovery is over, or at least, confined to the deepest parts of the Amazon rainforest or something.
Or the bottom of the Mariana Trench.
Exactly. We certainly don't expect to find uncharted territories sitting directly adjacent to our own driveways.
No, not at all. But the moment you actually stop walking and you kneel down in the dirt to look closely at the underbrush, that beautifully printed encyclopedia suddenly looks severely incomplete.
Oh, totally.
You realize we're actually surrounded by an incredible amount of biological dark matter, just uncategorized life hidden in plain sight.
Hidden right there in the backyard.
Right. And that brings us to the mission for today's deep dives.
Hey,
We are exploring a literal backyard mystery that proves the era of discovery is very much alive.
it's just wild.
It is. We are taking you on a journey today to unpack a singular, deeply intriguing field note from a gardening journal.
Yeah, and it's a remarkable piece of grassroots documentation. We are analyzing a post by an author named Ang.
Published on April 23, 2026.
Right, and, the context of this publication is absolutely vital. It is specifically titled Notes on Gardening, Part 874, Evaluating an Unidentified Plant Species.
Okay, let's unpack this. Because Part 874 is easily the most intimidating yet exciting part of this entire source material.
Oh, without a doubt.
If you are on Part 874 of your gardening notes, you are not some weekend warrior.
No, you're not just someone who bought their very first Monstera plant at the local hardware store.
Exactly. You have put in thousands of hours of observation.
Yeah, the discipline required to log 874 separate observations implies a massive, systematic data set.
It's huge.
Ang is someone who has tracked complex phenological cycles, watched microseconds change, and cataloged how local flora responds to shifting weather patterns over an extended period.
This is rigorous, long-term citizen science,
Spot on. It really is.
which is why I look at this scenario like watching a long-running TV detective show. You know the exact trope.
Oh, yeah, the seasoned inspector.
Right, the seasoned, seen-it-all inspector who has worked 1,000 cases. They know the geometry of the streets. They know all the usual suspects by name.
They've seen everything.
But then suddenly they walk onto a crime scene that just shatters their entire paradigm. The geometry is wrong. The clues make no sense.
Yeah, that is Ang here.
Ang notes in the journal that they've compared this plant to, quote, numerous others, and they're still completely stumped.
And that perfectly establishes the stakes of this specific mystery. Because, if an observer with hundreds of methodical journal entries under their belt doesn't recognize this plant, we are dealing with a genuine botanical anomaly.
A real mystery.
Yeah. But what is truly important here is how Ang reacts to that anomaly. Ang specifically writes that they haven't quite managed to pin down a clear identification.
Right, they don't try to force a label onto it just to feel a sense of closure.
Precisely. A lesser observer, someone desperate to maintain the illusion of that finished map we talked about, they might have just misidentified it.
Oh, sure. They'd say, oh, it's just a common weed with a weird mutation.
Exactly. Or they'd ignore it entirely because it didn't fit their mental database. But the true mark of scientific expertise is the active willingness to admit when you simply do not know.
To just embrace the ignorance.
Yeah. Ang treats the unknown not as an annoyance, but as a formal subject of study.
It is the anomaly that actually drives the scientific method forward. So let's examine the physical evidence Ang found at the scene of the crime.
Let's do it.
The journal provides a very specific morphological description. The plant features, quote, small, elongated leaves with a distinct white stripe down the middle.
Right. And based on that single prominent physical trait, Ang immediately applies deductive reasoning. Ang notes in the journal, I'm assuming it's a variegated variety.
Here's where it gets really interesting, because going back to my detective analogy, that distinct white stripe is essentially a suspect sketch.
Or a unique fingerprint left at the scene.
Exactly. But a fingerprint is only useful if you have a database to run it against. So I am looking at this and wondering, how much can you really deduce from a simple lack of pigment down the center of a leaf?
That's a great question.
Does that actually narrow the field in any meaningful way?
It narrows the field drastically, actually. What's fascinating here is the underlying biological mechanism of what Ang is categorizing.
Okay.
Because variegation in botany isn't just a cosmetic paint job, it indicates a fundamental cellular condition.
What do you mean?
Well, it often points to a genetic mutation, specifically a chimerism where two different chromosomal makeups are coexisting within the same plant tissue.
Wait, really? Two different makeups in one leaf?
Yeah. The green parts of the leaf have normal chloroplasts, but that distinct white stripe indicates a localized area where the cells literally lack chlorophyll.
Wait. If that white stripe is a localized lack of chlorophyll, then that inherently changes how the plant functions, right? Because chlorophyll is what drives photosynthesis. So if this mystery plant has a large white stripe down every single leaf, it has significantly less surface area dedicated to converting solar energy into sugars.
You have hit the exact metabolic consequence of variegation.
Wow.
Yeah. By identifying the plant as a variegated variety, Ang isn't just saying, well, it looks like these other plants. Ang is categorizing its physiological behavior.
Oh, that makes so much sense.
This plant is operating at a permanent energy deficit compared to a solid green counterpart.
So it's metabolically compromised right out of the gate.
Exactly.
So even without knowing its exact taxonomic name, Ang has successfully built a behavioral profile of the suspect. They know it will process light and energy differently than all of the surrounding flora.
It moves the mystery from the realm of the completely unknown into the realm of the conditionally understood.
That's brilliant.
It is. And that metabolic clue perfectly bridges into the next phase of the journal entry, the transition from merely observing the plant in its wild habitat to actively cultivating it.
Because an energy-deficient plant out in the wild is, well, highly vulnerable, which practically begs for scientific intervention.
Yeah, you have to step in.
Right. The journal notes that Ang, quote, harvested a sample for evaluation in a potted environment.
Yeah.
And Ang prefixes that by writing, as usual. Meaning this is standard operating procedure for Inspector Ang.
Right. You find a suspect, you bring them into the precinct for a controlled interrogation.
Exactly.
It is the critical shift from in situ field observation to ex situ controlled cultivation.
Right, because out in the wild, the plant is subjected to countless chaotic, uncontrolled variables.
Oh, yeah. It is competing with invasive root systems for nutrients. It's facing unpredictable herbivore predation, and it's dealing with, fluctuating soil moisture levels.
It's a war zone out there.
It really is. By moving a sample into a sterile potted environment, Ang is isolating the botanical variable. They're stripping away all that ecological noise to see how the plant performs strictly on its own merits.
They're locking down the variables. But the text notes that while the plant has, quote, acclimated well to the pot, its growth is proceeding at a, quote, sluggish pace.
Right, the sluggish pace.
And I have to admit, when I first read that, my instinct was to push back against the methodology. So what does this all mean?
How so?
Well, if you rip a wild plant out of the complex ecosystem it germinated in, sever half its root system, and shove it into a constrained pot, isn't sluggish growth just a textbook symptom of transplant shock?
That's a very fair point.
I mean, when my houseplants get sluggish, it usually means the root hairs are damaged and the plant is completely failing to uptake water.
It is a highly logical assumption. And honestly, it is exactly the trap a novice gardener would fall into.
Right.
A novice would see that ex situ sluggish growth, assume their cultivation technique was failing, and immediately try to correct it.
By doing what? Like adding fertilizer?
Exactly. Aggressively altering the soil chemistry with heavy fertilizers or over-irrigating it. But if we connect this to the bigger picture, Ang avoids that trap entirely.
How do they avoid it?
Because of one crucial detail. Ang specifically notes in the journal that this sluggish pace perfectly matches what they observed while the plant was still in the wild.
Oh, which loops right back to our deduction about the variegated white stripe.
Yes, exactly.
Because of that genetic chimerism, because it lacks the chlorophyll necessary for peak photosynthesis, its natural metabolic rate is just inherently slower.
You got it.
The sluggishness in the pot isn't a symptom of transplant shock at all. It is just the plant's genetic baseline.
Exactly, and that highlights the absolute non-negotiable necessity of gathering baseline wild data before running a controlled ex situ experiment.
It's otherwise you're just flying blind.
Right. If Eng hadn't spent the time meticulously logging the plant's natural rhythm out in the field, they would have completely misinterpreted the results in the pot.
They would have tried to fix a problem that didn't exist.
Yes, they would have introduced new, chaotic variables to cure a metabolic disease that the plant simply did not have.
Man, it is a masterclass in observational restraint.
Mm.
You have to trust your initial field data over your own anxiety that you're somehow, failing as a cultivator.
It bridges field ecology and controlled botany just seamlessly. The wild observation informs the controlled experiment, and then the controlled experiment confirms the wild observation.
And Ang clearly possesses the patience for this kind of long-term scientific feedback loop.
Definitely.
Because the final section of this journal entry outlines a highly ambitious testing phase.
Yeah, the next steps.
Ang vows to keep this potted sample under, quote, close observation, but also states, given the opportunity, I'll harvest a few more samples from the wild to get a better sense of how it performs under different microclimates.
This raises an important question. What is the actual scientific objective of moving beyond the single pot?
Okay.
By deliberately introducing new samples to different microclimates, Ang is elevating this project from basic cultivation to a rigorous multivariable stress test.
I was conceptualizing this like a botanical space
Oh,
program.
I like that.
You have your initial successful launch, the first potted plant that acclimated well under controlled baseline atmospheric conditions on Earth.
Right.
But now, Ang is planning to send a new batch of astronauts, these additional wild samples, to entirely different planets in the backyard.
That's a great analogy.
And we really have to extend that analogy because the spacesuit these astronauts are wearing, those variegated leaves with the white stripe, has a known structural vulnerability.
The lack of chlorophyll.
Exactly. If you send an astronaut with a faulty radiation shield to a planet with intense solar flares, they are not going to survive the exposure.
No, they won't.
Similarly, if Ang puts this energy-deficient plant into a microclimate with aggressive, direct afternoon sun, those white stripes lack the protective pigments to prevent phototoxicity. The leaves will literally burn.
The extension of that analogy holds up perfectly. And conversely, if Ang places a sample in a microclimate with heavy, persistent shade, the plant might simply starve.
Because it can't get enough light.
Exactly. Its reduced chlorophyll capacity can't capture enough of the sparse ambient light to sustain basic cellular functions.
Wow.
But what makes this master clan so robust is that microclimates are defined by far more than just photon exposure.
Right, a microclimate is an incredibly complex web of localized gradients.
Oh, absolutely.
We're talking about variations in soil pH caused by decomposing leaf litter under a specific tree, or dramatic differences in ambient humidity trapped against a brick retaining wall versus an open, breezy patch of lawn.
There are even invisible subterranean variables at play.
Like what?
Well, one microclimate in Ang's yard might feature a robust mycorrhizal fungal network in the soil that the plant's roots can tap into for supplemental nutrient exchange.
Oh, fascinating.
Meanwhile, a sterile, sandy microclimate just 10 feet away might force the plant to rely entirely on its own sluggish metabolic processes.
It's a total gauntlet.
It is. By exposing genetic clones of this mystery plant to this gauntlet of diverse microclimates, Ang is mapping out the absolute phenotypic plasticity of the species.
They are mapping its evolutionary boundaries.
Yeah.
They aren't just trying to figure out what keeps it alive. They are actively searching for what ecological pressures cause it to break down.
Exactly.
If it survives the acidic soil but dies in the alkaline soil, that tells you something profound about its evolutionary history. It provides a massive clue about where this plant might have originated before it mysteriously ended up in Ang's line of sight.
It is the ultimate expression of the scientific method, executed without a multi-million-dollar university laboratory.
Just in a backyard.
Yeah. This level of comprehensive ecological profiling is happening through simple terracotta pots, native soil, and the sheer power of human curiosity, carefully documented in entry 874.
It makes you wonder, how many of the commercial agricultural staples or common houseplants we rely on today started as an unnamed variegated mystery in a citizen scientist's journal 100 years ago.
Probably a lot of them.
Right. The meticulous process of observing an anomaly, isolating its variables, and stress testing its microclimate tolerances. That is exactly how humanity has domesticated the natural world.
We are reading a snapshot from April 2026, but the methodology Eng is employing is ancient, it's foundational, and it's incredibly rigorous.
So, to bring this all together, let's summarize this incredible journey contained within this single field note.
Let's do it.
We started with a solitary observation that defied the accumulated knowledge of 873 previous entries.
Yeah.
We used the morphological fingerprint of a distinct white stripe to deduce a plant's metabolic deficit.
We saw how crucial wild baseline data is to properly interpret ex situ sluggish growth, preventing the false assumption of transplant shock.
Exactly. And finally, we unpacked a highly ambitious plan to test an unknown species' evolutionary limits across a gauntlet of shifting microclimates.
It serves as a powerful reminder that critical thinking, hypothesis testing, and rigorous deduction, while they're not constrained to sterile environments or academic institutions,
Not at all.
they are active, vital processes happening right now in the soil of ordinary gardens, driven by passionate people who just refuse to ignore the unknown.
Curiosity is a muscle, and Aang is clearly putting in the reps every single day.
Absolutely.
Which brings us back to that encyclopedia we talked about at the very beginning of our deep dive. The comforting illusion that the map of our local environment is completely finished.
Right.
But if we consider the broader implications of what Aang is doing with those microclimates, we have to acknowledge a much larger reality. Our global macroclimate is rapidly changing.
It is. The baseline environmental data is shifting globally, and it is happening at an unprecedented rate.
Exactly. The predictable microclimates in our own backyards, the historical frost dates, the ambient humidity levels, the average soil temperatures, they're altering faster than traditional botanical institutions can track them.
They really are.
Which means grassroots citizen scientists like Aang, meticulously logging their 874th entry, aren't just engaging in an eccentric gardening hobby.
No, they might actually be the essential vanguard of modern botany.
Yeah, they are documenting real-time ecological adaptations and migrations that the rest of us are simply walking past on the sidewalk every single morning.
Just completely missing it.
Right. So to everyone listening, the next time you step out your front door, ask yourself this: what vital shifting mysteries are happening right in front of you, just waiting for someone to stop, kneel in the dirt, and give them the attention they deserve?
The map isn't finished. In many ways, it's actively being rewritten.
You just have to be willing to read the leaves.